I am truly delighted to welcome all of you to Science Meets Music 2022. This is really it, okay? And I have to say, not being together for the last 2 years, that greeting comes from the heart, okay? It is fantastic seeing a number of you that haven't seen for the last 2 years. For those who don't know who I am, I'm David Fitzpatrick, CEO, Scientific Director, Max Planck Florida Institute for Neuroscience. And it is such a pleasure to be back here in this beautiful auditorium in the Benjamin School, celebrating science and enjoying an evening of wonderful music with you. And to make it even more special tonight, I'm happy to announce that we are joined by our good friends from Germany, Nobel laureate and Max Planck director, Dr. Stefan Hell, and his lovely wife, Dr. Anna Hell. Great to see you, and thank you for coming. Well, speaking of celebrating, this year we have something very special to celebrate. It's been 10 years, believe it or not, since we opened the doors to our institute over on Donald Ross Road, inaugurating the one and only Max Planck Institute in the United States, an institute dedicated to understanding the complex neural circuits that make up our brain. And, not to brag, but I'm very proud of our accomplishments in the last 10 years, including over 175 discoveries that have been published in high-impact scientific journals, changing our understanding of the brain and establishing Max Planck Florida as a world-class research institute at the cutting edge of neuroscience. Not bad for 10 years, okay? But thank you. But in addition to our high-impact discoveries, another achievement that we're very proud of is having created an environment that provides young scientists with freedom to engage in curiosity-driven discovery, where creative ideas are paired with state-of-the-art technologies, leading to discoveries that launch highly successful independent research programs. Using this approach, in the last 10 years, Max Planck Florida has helped launch the careers of 20 neuroscientists who are now faculty at distinguished universities in the US. I'm sorry, it's breaking it up. And around the world. This is, a scientific family tree that extends far beyond Jupiter and promises a future full of discoveries with profound impact on the field of neuroscience and our lives. And that success has also helped us recruit 5 new exceptional research group leaders to Max Planck Florida in the last few years, including Dr. Sarah Stern, who you'll be hearing from tonight and whose research is focused on something that impacts everyone in this room and probably everyone in the world. Sarah studies the neural basis for feeding behavior, what drives us to eat, whether it's biological hunger, boredom, or habit. She's working to understand how the brain sends signals that drive us to eat, and how these brain circuits are altered in devastating conditions such as eating disorders. Now, even though 20 million Americans struggle with an eating disorder or related condition, relatively few scientists focus on this area of research, in part because the level of federal funding is far less than for other brain-related disorders. As you will see, we are extremely fortunate to have the Stern Lab at Max Planck Florida. And because of the support she receives from the Max Planck Society and donors and supporters like you, she is able to continue the critical research with state-of-the-art technologies. Now, being able to focus on an important, challenging, fundamental question is what makes the Max Planck Institute such a special place to pursue high-impact science. And we want to thank you so much for your support of curiosity-driven research and for investing in a better future for all of us. I'm applauding for you, okay? Thank you. Now, is Dave Valse with us? Okay. Well, I'm going to thank Dave Valse. He is the head of school for the Benjamin School. And Benjamin has been such a wonderful partner for our series. And we really thank them. And if he were here, I'd be thanking him up and down, allowing us to have the event in this wonderful auditorium. So, with that, how about some music? All right. So, tonight's musical guest is pianist Robin Arrigo. Dr. Arrigo is a Yamaha artist, concert pianist, collaborative artist, and adjunct professor at Palm Beach Atlantic University. Her extensive musical experience includes performances throughout South Florida, New York, and Europe, and she's been a guest soloist with Palm Beach Atlantic University Symphony numerous times. Other concert venues include Kravitz Center, Palm Beach Symphony, Flagler Museum, Bethesda by the Sea, and the Juilliard School, just to name a few. She studied and performed abroad as both soloist and collaborative artist with Fondation Belle Arts Paris International Summer Session under the tutelage of well-known artist and piano pedagogue Nelson Delvini Fabri. And I encourage you to check out Robin's website, and you're going to find a wonderful quote from Plato that I had not seen before. Music gives a soul to the universe, wings to the mind, flight to the imagination, and life to everything. I think that says it all. Join me in welcoming Dr. Robin Arrigo. That was absolutely beautiful. Thank you so much, Robin, for that. So, now it's time to introduce tonight's speaker, Dr. Sarah Stern. Dr. Stern started her research group leader position at the Max Planck Florida Institute for Neuroscience January 2021, so she's a newbie. And she formed the research group leader called Integrative Neural Circuits and Behavior. Her research focuses on understanding how learning and innate behaviors combine to impact behavior. And as I said earlier, one of the areas that lies at the heart of her research program is understanding the neural basis of feeding behavior. She received her undergraduate degree from New York University and earned her PhD from the Icahn School of Medicine at Mount Sinai in the laboratory of Dr. Cristina Alberini, where she studied memory formation, consolidation, and enhancement. She then went on to become a postdoctoral fellow at Rockefeller in the laboratory of Dr. Jeffrey Friedman, where she first brought her expertise in memory and rodent behavior to the study of feeding behaviors, exploring the brain pathways through which environmental cues lead to overconsumption. And Dr. Stern also led a project studying the intersection of stress and feeding behaviors relevant to anorexia nervosa. I'm sorry, Sarah, I see Dr. Stern here, but I want to say Sarah, I'm sorry, is the recipient of numerous honors, including a K99R00 Pathway to Independence Award and a NARSAD Young Investigator Award. Join me in welcoming Dr. Sarah Stern.
Okay. Hi, everyone. I'm Sarah. Thank you so much for being here tonight. It's such an honor to be here in a really beautiful venue and beautiful music as well. So I feel really fortunate to be able to tell you a little bit about the research that I've done during my postdoc and that we're starting to do in my new laboratory. So as you've probably read, the title of the talk is Nature, Nurture, and Nutrition, Brain Circuits Underlying Obesity and Eating Disorders. So I'm gonna start the talk actually with a picture, and this picture, well, maybe, no, okay. Okay, so this picture is from a clinical report that was published in the Journal of Clinical Investigation in 2002. And as you can see, it's a picture of two boys. And I want to start by asking, if I were to ask you what the difference between these two boys is, most of you would probably say that the boy on the left probably eats more than the boy on the right. And if you were to say that, you would be correct. If I then further asked you why does the boy on the left eat more than the boy on the right, I might then get a number of different answers. So some people might say, well, maybe the boy on the left doesn't have enough self-control surrounding eating, and the boy on the right does. Or some might say, maybe the boy on the left is in a home in which there are too many snacks available and doesn't live in a healthy eating environment, and the boy on the right is fed nutritious, healthy meals. Or you might suggest that maybe there is some underlying biological issue that leads to this difference in eating behavior and in weight between those two. And I'm going to tell you more about it after. In fact, there is a biological reason that the boy on the left eats more than the boy on the right, and in fact, it is the same boy. But first, I'm going to tell you a little bit about what we know about how our body and our brain controls feeding. So this is the canonical model of what we call homeostatic feeding, how your body is able to regulate its weight on a day-to-day basis. So for example, right now, I haven't eaten dinner yet, and it's dinner time, so I might be hungry. And in that case, you have a certain amount of fat tissue. Everyone has fat tissue. That fat tissue releases a hormone that was discovered by my former mentor, Jeffrey Friedman, and that hormone is called leptin. Leptin sends a signal to a part of your brain called the hypothalamus, which tells your brain how big your fat mass is. And so once you go and you find the food that you're looking for, you're feeling nice and full, your fat tissue expands, and as your fat tissue expands, it starts to release more leptin. And that increase in leptin sends the signal to the hypothalamus that you're no longer in need of additional calories and you can stop seeking food. Okay, so in fact, if we go back to these two boys, they are, as I said, the same boy, and this boy was born without this gene leptin. And because he was born without leptin, he has no signal that comes from his fat tissue to tell his hypothalamus that he no longer needs to eat. And so he feels in a constant state of starvation and is always seeking food. We can also recapitulate this in the lab. In fact, the gene leptin was discovered in the lab. And so these are two mice, and just like the boy, the mouse on the left is missing this gene leptin. We call this the OB mouse. OB stands for obese. And the mouse on the right, his friend, has a normal functioning leptin gene and therefore is of normal weight. So the problem is that it turns out that these single gene defects that lead to obesity only account for about 10% of cases of obesity worldwide. And in fact, leptin deficiency has only been found in about 100 patients total. So the question is, where are the other 90% of cases of obesity coming from? And so in reality, it turns out that variation in body weight is not just due to genetics, so that's what we call the nature component, right? It's something you're born with. It's due to this complex interaction between your genes, or nature, and your environment, or nurture. And so by environment, we mean lots of things, right? It's like the food that's around you, it's the culture, it's cues in the environment. And so this is what my lab aims to study, and what I've been studying for a number of years now, is how these environmental factors actually interact with these innate feeding circuits to cause these changes in feeding behavior and ultimately in weight. And so in particular, we study how cues in our environment can end up leading to overconsumption. And so on the next slide, I'm going to show you a clip to demonstrate what I mean by this. So this clip comes from The Office. For anyone who hasn't seen The Office, the guy in the blue shirt is Jim. The guy in the suit jacket is Dwight. And Jim likes to play a lot of tricks on Dwight. And he's going to use this phenomenon that I'm going to talk about, which is classical conditioning, to change Dwight's behavior. Okay, so you can see that cues in our environment, they are potent regulators of our behavior, and they can actually change how much we eat. And these cues are all around us, right? So the most famous example are the golden arches of McDonald's. They induce cravings for Happy Meals and burgers. Kids are often targeted by this type of, food cue with mascots on foods that companies want you to buy for them. But context can also have the same effect, right? So for example, in the US, most children are conditioned, maybe not during COVID, but in general, to go to the movies and eat popcorn, right? It's something we learn when we're very young. But if I brought you to a symphony or, classical music event like we're at tonight, and I gave you a bucket of popcorn, you'd probably be much less likely to, dig in and eat it, because we are conditioned to understand that in one context, we should be eating this like salty, crunchy, great snack, and in the other context, we should not. And so we use this type of behavior to try to understand how these seemingly neutral cues in our environment can actually promote this type of overconsumption or binge-like behavior. And so my expertise is in rodent behavior, so we design tasks that utilize these different aspects of the environment. So we take these two different contexts, so you can think of the context on the left as the symphony. It's the context in which we are not going to give mice access to food. They're not going to learn any association there. And you can think of the context on the right as the movie theater. So it is the context in which the mice will have access to food, and they will learn to associate this specific place with food intake. And the arrows are just pointing to the place within the context where food may or may not be available at that time. So you can think of that sort of like a concession stand. Okay, so we take these mice and we put them in this movie theater context. They're hungry and we give them food there, so they want to eat. And by eating in this context, they start to form this association, just like Dwight formed that association with the ringing of the computer. And so in the graph on the right, I'm showing you how much these mice are eating. So the bars represent how much the mice eat in grams. So obviously the taller the bar, the more they eat. And so of course these mice are hungry, so over these 2 days in which they're forming this association, they're going to eat in this context. So then we can give these mice what we call our overconsumption test. And so we are putting them back into this movie theater context that they've now associated with food, or we're putting them back into this symphony context, which they have seen before but was never associated with food. But now we're going to give them access to food in both of these contexts, and we're going to see how much they eat. And so just the same way at the movie theater, you are much more likely to eat the popcorn than at the symphony, these mice do the same thing. So in this movie theater context, because they've been conditioned to learn that it's associated with food, they eat much more than they eat in the context that they haven't associated with food. And so we use this as a model to study how cues can induce binge-like behavior in these mice. And once we have the behavior, we can start to really look at the neural circuits and try to understand, how does the brain actually do this? How does it learn about these cues and make this association with food to actually override these homeostatic feeding circuits? Okay, so we focus on an area of the brain called the insular cortex, which is not a very well-known area of the brain, but it's sort of on the side, on the lateral surfaces, and from human imaging studies, we had some idea that the insular cortex tends to respond or be active in response to food cues, and particularly in patients that either suffer from obesity or eating disorders. So we decided to see if the insular cortex was important for this behavior. So I'm just going to show you also, so this is the mouse brain, and the arrow at the top, well, the arrow at the top and the bottom, they're both pointing to the insular cortex in mice. And one really interesting thing that we can do is that we can actually make a mouse brain transparent so that we can put different labels into a part of it and see what areas that part of the brain connects to. So in the video that I'm going to show you, on the right, I've injected a tracer into the insular cortex of a mouse brain, and what you're going to see are all of the projections that it makes. So this is the tracer that's labeling the insular cortex, which runs almost down the entire side of the brain. And then you can see that it projects across the brain to the insular cortex that's on the other side. And it also makes these strong projections to areas downstream, like the hypothalamus. And so through this projection to the hypothalamus is a potential mechanism by which this brain area might be able to override these homeostatic feeding circuits that I told you about before. So we wanted to know, if we take this brain area, the insular cortex, offline during this test, will we be able to prevent binge-like behavior? And indeed we do. So what I'm showing you here is that we can inject some substances that take this brain area offline. So I'm showing you on the sides the insular cortex. And then the blue bars, I'm showing you the same control group, right? So these are mice that we've trained to overeat in response to this movie theater-like context. But if we take this insular cortex brain region offline, we no longer see this binge-like behavior. But what I find really amazing is that if we take this brain area, the insular cortex, offline, and we look at how much mice eat just over a 24-hour period in their normal home cage where they live their whole lives, we actually see no difference in how much they eat. And so what this tells us is that we have a homeostatic feeding circuit that regulates how much we eat on a 24-hour basis, But the insular cortex really has nothing to do with that. Instead, it is responsible solely for integrating this information about the environment and using that learned information to control feeding behavior outside of these homeostatic mechanisms. Another thing that we can do and have done is that we can actually image the activity of these insular cortex neurons while mice are engaging in behavior. And we do this by placing a tiny little microscope lens directly into the insular cortex, and we can mount a miniature microscope on top so that we can directly visualize the neurons in real time. So this is an example of what that looks like. So this is a mouse who's been implanted with this microscope, and he is just running around an open field. And on the right, I'm showing you a field of view from this lens, and you can see that every time you see a pop of light, that is a neuron that is active at that particular moment. And so we can sort of aggregate the activity from all of these neurons together so that we can look at how they respond during this task. And so here I'm showing you this trace that is the aggregate of all of this neural activity in the insular cortex. And in blue, I'm showing you feeding bouts. So that is every time the mouse is actually eating. And so hopefully what's easily apparent is that every time the mouse eats, there is a really big increase in neural activity within the insular cortex. And so as this mouse is engaging in feeding, these neurons are active and encoding information about the environment. So what I want you to take away from this is this basic idea that you have these sort of two systems in your brain. You have the hypothalamus, which is like the nature system. So it controls this homeostatic feedback loop that, ensures that you maintain energy state over the day. But you have this second system, which includes the insular cortex and some other regions I don't have time to talk about today. And that is the nature system. And it's a separate system that integrates this information about the environment to override the hypothalamus and potentially change feeding behavior and potentially lead to conditions like binge eating disorder and obesity. Okay, so for the last few minutes, I will talk about anorexia nervosa. Anorexia, like obesity or binge eating, is also a complex disorder that involves both nature, so biology and genetics, and nurture, so the psychological and environmental factors. And yet, it's, as David mentioned before, severely understudied. So despite the fact that it has the highest mortality rate of any psychiatric disorder, and there are no FDA-approved drugs, it receives very little federal funding for research. And so the question is, why is that? And I would argue that there is a lot of stigma surrounding eating disorders, right? So a lot of people tend to think, so just like I showed you those two boys in the beginning, and I said, some people might say it's a lack of willpower or the environment. People tend to think the same thing about anorexia, right? That it is a disease that is due to fashion magazines, and people will say things like, just snap out of it, you just want attention, it's no big deal, just eat. And don't really acknowledge that it is a disorder that has a real biological component. But in reality, anorexia is not a new disease at all. It goes back as far as the 1300s. So St. Catherine of Siena is known to have suffered from anorexia nervosa. She actually died prematurely of malnutrition. This was called at the time like holy starvation or holy fasting. And in men, they called it asceticism when they saw it. But despite the fact that people were desperately trying to get her to eat, she refused and eventually died. Mary, Queen of Scots, also is known to have been very ill and underweight throughout her life, and historians now believe that she also suffered from anorexia nervosa. But the first documented cases of anorexia that were actually considered anorexia were still really early, like in the 1800s. And actually the first documented case of anorexia was in a young man. So again, not just a disease that's in women and definitely not a new disorder. It's also not just a disorder of willpower. So anorexia nervosa has a strong genetic component, and it shares a lot of genes with other psychiatric disorders, in particular with obsessive compulsive disorder. So on the left, I'm showing you correlations between genome-wide association studies where they look at genes underlying a variety of different diseases and personality traits. And you can see at the top, the highest correlation is with OCD. And surprisingly, at the bottom, you'll see that there's not a lot of correlation with metabolic disorders, with obesity. So anorexia does not seem to be just the flip side of obesity. It, in fact, shares much more commonalities with other psychiatric disorders. And so anorexia shares this cycle with OCD, again, showing their similarities, right? So they start off with anxiety and distress, and that leads to these maladaptive behaviors, so compulsions or restrictive eating. And that provides the patient with some temporary relief, but then it cycles back to these obsessions with food or with these compulsions, and again, leading to this anxiety and distress. So we know that anorexia shares a lot of these commonalities with psychiatric disorders, and yet there are really almost no animal models to study this disorder, although there are many models to study obsessive compulsive disorder. And so that is what my lab is really focusing on right now, is trying to develop an animal model like we did for binge eating so that we can start to identify what are the underlying neural circuits, and so that then eventually we can use that to design treatments. And so what we're trying to utilize is this idea of anxiety and distress. So we know that in patients with anorexia, symptoms are often precipitated by traumatic events or stressful events. And later on, cues that predict that type of stress will then trigger these restrictive eating episodes. And so we try to do that in mice. So we take mice, we expose them to a stressful event. So for mice, that's something like being placed in a very confined location for a short amount of time. And then we also expose them to food cues like we did for our binge eating task. But we pair those together so the mice start to associate not just the food cues with the food, but also stress with this whole food cue situation. And so then we can test what happens now when we expose mice to this food cue. And so in pilot experiments that we've done, we see that once we do these stress food cue pairings, if we place mice back into this context where they've had this food cue, they still engage in that binge-like behavior that I told you about before. So on the right, I'm showing you in red the mice that were exposed to stress, and they eat more compared to our control mice. But then when they go back to their home cage and we measure their body weight and food intake over a period of weeks, these mice will severely restrict how much they eat. And so we think this is a potential model for a binge restrict type of anorexia that's very common in patients, but has never yet been modeled in rodents to understand their neural circuits. And so our lab hopes that we can use this information to try to identify genes within these brain regions that are responsible for these behaviors so that we can design better treatments that will help treat these devastating disorders. So I just want to end by thanking all of my lab members, of course the Max Planck Society that is supporting my research, all of my colleagues at Rockefeller, especially my mentor, Jeffrey Friedman, and all of the funding that has supported this work throughout the years. We couldn't do it without them. So thank you very much. you.